Good morning. My name is Jillian Cristiracho, and this is my report. A canker disease of apple caused by Cytospora parasitica, recorded in China. Apples, or also known as Malus domestica Borg, its scientific name, are among the healthiest and most popular fruits in the world. Um, Apples are one of the popular fruits everywhere and are also nutritious. Among all crops, these also have a great value. Apple is also served as a model reference when it comes to evolutionary mechanism or processes for those clonally propagated perennial crops or that are grown in clonal. So, one of the most damaging disease in China's apple producing region is apple canker disease, which is brought on by Cytospora species. Wala lang. So, dagan ng species si Cytospora nga nakakos sa disease sa mga apples. So, main stems, branches, and seedlings are affected at this canker disease. This article focuses on the specific species of Cytospora, which is Cytospora parasitica. And this article also na 16 fungi species na main antagonist sa sakit sa apple trees worldwide due to Cytospora. Methods. Number one is sample collection. So, sample collection is collected samples from the surface of apple from the affected cankered to healthy tissue. Ang mga samples were kept individually in craft paper bags at room temperature. And fungal isolation. So, 5 trees in each location, 5 trees in each location and 5 cankers per tree were sampled to know the pathogen or the fungi. Ano nang iya? Butang diri. Bark pieces 5:5 mm were surface disinfected in 75% ethanol for 3 to 5 seconds and 0.1 mercury chloride for 1 minute. So, and using a dissecting microscope and sterile knife, a block of agar with a single hypal tip was aseptically removed from the edge of each isolate's culture to create pure cultures, which were then transferred to new PDA and incubated at 28 Celsius. All isolates were kept on new PDA's lands and kept at 4 Celsius. That's pathogenicity test. So, sa pathogenicity test, vitro-inoculation assays were carried out to identify the pathogens in the isolated samples. So to evaluate the isolate's pathogenicity, in vitro inoculation assays were carried out. So each of the 8 isolates from Fuji apple trees 5-day-old cultures had 5 mycelial agar plugs that were 5 mm in diameter. A 5 mm diameter cork borer was then used to remove a 5 mm lump of bark from the top of the branch. The injured tissue was plugged with a 5 mm diameter mycelial plug and covered with a parafilm. Next. Morphological characterization. So sa morphological characterization, the parts of the bark where fruiting process happens such as Picinidia, stromata, disc, osteolis, conidia, and conidiophores were viewed and photographed under a light microscope. In this method, the classification were created. Under a light microscope, pangsulti, the Picinidia, stromata, disc, sila. So, Leica DM750 light microscope, manang gigamit. DM750 light microscope was used to measure the length and diameter of 50 conidia from randomly chosen conidiomata of each isolate. So, lastly, molecular characterization and multi-leucos phylogenetic analysis. Total DNA was extracted using an ASOP column fungal genomic DNA purification kit in order to identify the isolates on a 1% agarose gel. The extracted DNA was evaluated for purity. Its 1 and its 4 primers were used to amplify the internal transcribes base or ITS region. The large ribosomal subunit was amplified using primers NL1 and NL4, and a transcription elongation factor region was amplified using primers AF1 688F and AF1 1251R. The annealing temperatures and primer pairs, utilized to amplify partial sequence fragments of the ITS, LSU, and EF sections and are provided. The Shanghai Envitrogen Biological Technology Company Limited did DNA sequencing at Beijing, China. Sigman 7 1.0 was used to put together the sequences, then compared with known sequences and GenBank by running a blast search. By bootstrapping 1,000 heuristic searches, and pylograms were created in Mega 6.0 using the neighboring joining method. Tabunan sa share screen. So, mauna niyan, this is symptoms. Bark tissue had a loose texture and a reddish-brown color during the early stages of infection. The lesion expanded during the course of the illness until eventually ceasing to grow, becoming somewhat erupted and dehydrated and sunken. Isolation and pathogeny tests. So 5 days following the injection, the branch's bark tissue shrunk and turned reddish-brown in color. Black dots were seen at the inoculation sites on the infected tissues 8 days after the inoculation, making it easy to discriminate between the lesions and the healthy tissues. On dead wood, there were round, erupent, black fruiting bodies. So for morphological characterization, conidial stromata, which were black disc shape, had a diameter of 89 to 162 basta di mabasa, and were shaped irregularly or round to oval when grown on PDA. All of the isolates colonies had a texture that were uniformly loose, whitish, and villiform in shape. The isolates produced black fruiting bodies after 25 days of cultivation, which were scattered randomly around the colony's exterior. Phylogenetic analysis. So the ITS, LSU, and AF sections were amplified using the primer pairs, yielding fragments of 560 BP, 580 BP, and 600 BP. There was 99% similarity between the sequences from the various strains. The newly discovered apple tree Canker in Xinjiang was determined to be a C. Parasatica by combining the outcomes of ITS and LSU gene fragment sequencing with morphological traits. So, discussion. The three main diseases caused by the Cytospora parasitica are canker, ring rot, and dry rot. This is the first case recorded of Cytospora parasitica infesting China causing canker disease, and it is also recommended to further conduct studies and if this Cytospora parasitica has also infecting other plants and to locate if this was brought naturally in China or was imported in other species. So I think that's all. And if you want to read more, nara dire. Kita. So, mga reference. Excuse me. So, para. Mawala to sa last. So, sa last din na itong giingon na lagi na pwede mag-conduct o study kung ang kanibang Cytospora parasitica nag-harm ba sa other plants or daya ba ni sa China naturally or na-import lang ba siya. So, that's all. Thank you.